Chinese society needs more medical diagnosis. What does that mean to the further development of PGM market? So our next speaker, Dr. Huang Lin, Professor Huang Lin, is going to talk about the challenges and opportunities facing or the medical industry is facing. She's from the Shanghai Institute of Thoracic Oncology. Honorable guests, ladies and gentlemen, good morning. Many thanks to Anglo America for inviting me to this seminar. My name is Huang Li. I'm from the Institute of Thoracic Oncology of Shanghai Chest Hospital. Today I'm going to talk about PGM-based in vitro diagnosis. So as China's population keeps increasing year on year and the number of people at or above the age of 60 is increasing as well as people's living standards keeps increasing, people's average life has also increased. As a result, the aging problem is all the more exacerbated in China. An increasing population of senior citizens mean that there will be increasing demand for medical resources. In recent years, China's medical spending has also increased year on year. We have always maintained spending at around 8%. In recent years, average disposable income of people have also increased as people are increasingly health aware and as people's spending on healthcare increase compared with advanced countries. However, the average spending on healthcare in China still lags behind. For example, in the United States, compared with the United States in 2016, average spending on healthcare in China was only 36% that of the US citizens. In other words, there's huge space for further improvement in China on this front with great potential. Of course, we've seen some favorable policies keep pushing forward the development of big healthcare industry in China. As early as 2011, there was a government initiative to build such a greater healthcare industry with new type of health products. And in 2016, there was a Health China initiative and campaign proposed at the very top level. With these favorable policies, China's healthcare industry is also facing structural changes. In the past, among all the healthcare companies, the pharmaceuticals were at the very top of the industry. They also represent the most profitable sector among all the healthcare sectors. However, with policy guidance, pharmaceuticals is facing increasing competition, and it is hard for them to find additional breakthroughs given some of the current conundrums that they are facing. And there are some restrictions on import replacement as well. So with these things, with these policies in place, IVD as a very important method for clinical diagnosis become very important method for clinical and identification as well as diagnosis. As a result, these traditional medical or healthcare devices companies find themselves having new opportunities thanks to years of education and promotion. People are increasingly aware that early-stage cancer has much higher cure rate compared with mid-stage or late-stage cancer. Let's just take gastric cancer as an example. If gastric cancer is identified at early stage, 95% of survival rate for 5 years, so whereas if it's late and mid-stage, 5 years survival rate of gastric cancer patients only 10%. So the gap is just very huge. According to some calculation, every year, 18 million cases of cancer is reported. New cases of cancer. And for China, the early screening market of cancer exceeds 40 billion RMB. In other words, IVD market is just huge. So let's take a look at China's IVD market share. So the IVD agent accounts for 82% of the total IVD market. And in China, most of the IVD companies are in the lower end of the market. They look at the biological, chemical, and they develop some immunity-related IVDs. So the lower end side of the market is in the cutthroat competition situation, whereas the higher end molecular diagnosis is dominated by overseas companies, including Roche, as well as other high-end overseas companies. So now we've seen new method and technology for in vitro diagnosis. You may have seen this, or you may have watched this film called Bad Blood. It actually represents some sort of reproduction of some real-world cases from Silicon Valley. They claim that they could use 1 drop of blood to do testing, and the process is in vitro and is non-invasive, and the testing results is very precise. So the concept is almost perfect. As a result, venture funds chase around the company. As a result, the company was valued $9 billion. Actually, the company's underlying technology relies very much upon the development of a low-end diagnostic test device. However, it's desktop-based. So if you use 1 drop of blood to detect over 200 biomarkers for disease, this is impossible. And the reality has sunk in, and the founder of this company was put into prison. However, this case still told us that there are big problems with regard to in vitro diagnosis of disease. Patent or PGM can bring new opportunities for in vitro diagnosis, as we know that IVD agents have 3 important characteristics and properties. The first being sensitivity, and second, specificity, and thirdly, it's stable. In terms of sensitivity, as was mentioned by previous presenters, platinum or PGM have very good catalytical activity. They can amplify the signal. As a result, it will be easier for us to detect biomarkers in any organism. And in terms of specificity, as PGM are easy for surface modification, so we can do specific recognition by molecular probes with molecular surface modification. Regarding stability, PGM have such properties as high melting point and stable properties, so it's very easy to turn them into reagent and for longer term storage. So this slide showed you what we have done over the past couple of years at our project team. We have done something and made some achievements on the part of IVD. We were focused on the development of chip-enhanced optical sensing in order to do single molecular level diagnosis of disease at very low cost, high throughput, and fast speed. For this slide, I'm going to give you more detailed introduction of what we have done based on platinum and some of our projects and research projects based on platinum. The first is platinum-based nanoreactor that's used for the diagnosis of pancreatic cancer, which is known as the king of all cancers, because pancreas is in a hidden place in the duodenum of the human body. Usually it's really hard for either ultrasound or color ultrasound to identify any changes on the part of the pancreas. So when pancreatic cancer is identified, it's usually in the late or mid stage. However, there are 2 characteristics with regard to pancreatic cancer. First, it causes metabolic disorder, and the second manifestation is differential glucose expression. So based on the 2 manifestations, we designed 2 diagnosis platforms based on platinum for POCT fast screening as well as precise molecular diagnosis. So we created this platinum nanoreactors for the catalytic reaction because it has very high catalytic efficiency. It can complete the testing within 1 minute with high specificity. It can specifically detect the existence of trace glucose inside human body. In addition to that, we also use platinum for enhancing the signal of mass spectrum. As you know that inside human body, there are high concentration of salt and protein. Usually, if we use other materials, due to the existence of high concentration of salt and protein, we cannot detect the biomarkers of disease that we want to see. However, based on the platinum-based spectrometry, you see very good properties of being salt resistant and protein resistance. This is the glucose curve that we built based on the platinum nanoreactor. You see that OD is as low as 39.126 nanogram per UL, and the process can be done within 2 minutes. In other words, it's fast screening, POCT, or on the part of the bedside. And also we have pancreas model to do comparison of a diseased pancreas with a healthy pancreas. We also see the blockage of the starch and sugar metabolic pathways that may become another target for the treatment of pancreatic cancer. We published our work on Matter, which is a sister publication of Cell with the influence factor of 19.967. We got recognition from some academician from the Chinese Academy of Science called Dr. Peng Xiaojun, and we have successfully transferred 6 patents after its research results, and based on the patents, 7 IVD reagent products were developed, and the second product is a platinum-based alloy for the screening of gastric cancer. As I mentioned, that gastric cancer is usually done for high-risk patients when they feel ill with their gastric part, and usually when you don't feel sick, you usually don't apply for any gastric probe diagnosis because first of all it's invasive, and second the entire process requires long-term preparation with your abdomen having no food inside, and it requires longer-term anesthesia. As a result, patients' compliance is low. That's why in China the screening efficiency of gastric cancer is always lower than many other countries. In terms of the serum-based detection or testing, there is no specific biomarker that's specific to gastric cancer, so there is an urgent need for us to develop a effective testing method for the early screening of gastric cancer. So in this project, we developed a material which is a platinum-based polyanion. It has very good porous structure. As a result, it has some size exclusion effect, which will facilitate the separation and pre-concentration of target analyzed from complex samples. So we shift our focus to alloy because compared with single metal, alloys have better physical and chemical properties, and as you can see that the size are of uniform size and the porous size around 2. So with such porosity and such structure with laser, it can create enhanced regional field of electricity compared with single unit of metal. The enhanced times can be 2 orders of magnitude. So this is something that we've done based on platinum-based polyalloy, and we get the metabolites, and that's the result for early screening of gastric cancer. As you can see, A and B stage 1 gastric cancer both come from stage 1 gastric cancer. For stage 1 gastric cancer, the precision rate of detection is 97%, far exceeding the existing cancer biomarkers such as CEA, because CEA has this problem in that it's broad spectrum biomarker. In other words, CEA increase may not necessarily come as a result of gastric cancer. Other types of cancers can also result in increased CEA, but we have also invited 3 director-level physicians to help us with the reading of the results for the same patient, and base their conclusion on the stain. Here on the chart here, you can see that the reading sensitivity or the reading specificity above a very high threshold. In other words, whenever they find this lesion, it's easy for them to detect whether it's malignant or it's benign. However, its sensitivity is not very high. In other words, it's really hard for them to identify the target or the lesion. So this is the slide, or this is the image, and the yellow arrow points to the disease area. Only 1 out of the 3 physicians detected as cancer, as opposed to our platform, which gave a definitive conclusion that it is a cancer. And we did some additional testing confirming the result. As for the imaging results, all 3 believe that it's not malignant. Only our platform, our material believe that it's malignant, and pathological results support our result. So the paper was published last year on advanced materials, and currently we have completed 100K level cohort validation, and currently they serve 23 tertiary hospitals in China. Next, we applied this polyanoid based on platinum to the stratification of diabetic retinopathy risk, because diabetic disease is something that occurs mostly to those who enjoy a relatively high living standard. And in China, the number or the proportion of diabetic patients increased from 1% to 11%. Out of those who have diabetic diseases, retinopathy is one of the most fatal consequences, and early screening of retinopathy can reduce its occurrence rate in 90% of the cases. However, we don't have very good hematological method. We only rely on, or we mostly rely on, the clinical experience of doctors plus imaging for the conclusion. So we're mostly concerned with two questions. How do we select the patients out of all the healthy population? And second is whether we need to put all the population through the expensive molecular spectrometry. So based on this, we developed this platform. One is a platform for coloring or for colorimetry to do direct screening of high-risk population. Out of the high-risk population, we put them through spectrometry to identify they have retinopathy or not, in order to achieve this differentiated pricing and clinical testing. So we have this platform allowing people to use their mobile phone to directly read changes in the concentration and level of glucose, as platinum polyanoids have such advantages as having very uniform bulk material co-crystallization, so ensure the repeatability of the testing result. And our platform, our colorimetry platform, is 4 times better regarding sensitivity compared with commercially available testing reagent, be it sensitivity or specificity. Two numbers both exceed 90%, and we apply them, or we integrate them with our mobile phone-based app. In other words, we can achieve simultaneously quantification and differentiation of glucose patients, as well as the diagnosis of smart retinopathy. So this result was published this year on Biosensors and Bioelectronics, having received extensive attention from relevant stakeholders. We also won top prize in some important event. Thanks again to the hosts, to Anglo America, for giving me this very wonderful opportunity to give you a presentation of our research results. Also, many thanks to the team members. Because of their perseverance and because of their hard work, we managed to turn our dream into reality. So basically, what we do is we use pattern-based nanomaterials, combining that with clinical design, and we integrate that with multimodal data so that we are able to identify diseases wherever they are. Thank you.